It's my pleasure to introduce today Max Scholar. Max got his bachelor's degree in molecular biology at Princeton, and he was during his time there, he was also part of a integrated science program in quantitative and computational biology, and you'll see that theme throughout his work. During his time at Princeton, he was also an undergrad researcher with David Botstein, a prominent and renowned genome biologist and genomics pioneer. For his PhD, he went off to Harvard and worked with Angela DePace, where they combined reverse genetic tools and systems of computational biology to understand how transcription factors work as enhancers to control gene expression in space and time in the Drosophila embryo. And now he's currently at the University of Washington, St. Louis, working with Barack Cohen. He has won a number of fellowships and awards over his time. Right now, as a postdoc, he has had a American Cancer Society postdoctoral fellowship. He's currently a Burroughs Wellcome Fund postdoctoral fellow, and he also holds a K99 R01, which will facilitate his transition to a faculty position. And one of his most awards that he says he's most proud of is his award for outstanding mentorship and teaching that he won last year as a postdoc. Just about his research really quick. So Max's current and future research is using high throughput quantitative assays combined with computational modeling to understand the biological logic that underlies the organization and structure of transcription factor activation domains. And the integrative approach that he's going to tell you about today has already yielded some significant, I think, in my opinion, some of the most significant insights into this domain organization since they were first discovered over 30 years ago by Mark Patashny and colleagues. So really interested to hear about that and also to hear about the future.
Thank you. Hi everyone, can you hear me?
Yes.
Great. All right. I'm really thrilled to be here. I've had a fantastic day. Thank you for that introduction. So today I'm going to tell you about some of my work and a glimpse of the plan for the future. All right, so I'm interested in transcriptional systems. Each of us starts life as a single cell with one genome. And through the really amazing process of development, we come to have all the different types of cells in our body. And we know that transcriptional regulation is really important for this process. Our fat cells have a different transcriptional program from our blood cells or our skin cells. And that how this transcriptional program is deployed during development is really essential for patterning differentiation and development. It's also very important in homeostasis. Our liver cells divide much more quickly than our neurons. But our neurons are also turning over all the channels in the synapses, and that's also a transcriptional process. So transcription is important in development and homeostasis. It's also important in stress response. So this is a Arabidopsis before drought, and this is after drought. And the wild type plants are able to survive this drought stress because they can mount a transcriptional program. And this MAX2 mutant has a defective transcriptional program, and the plants have a much lower survival rate. And as many of you know, when transcription goes wrong, it can cause all manner of disease. It can cause really bad developmental diseases, but also things like cancer. This is a melanoma. Transcriptional regulation and transcription factors are also important for evolution. And so this is an example of behavioral evolution within species. The DMRT3 gene, there's a truncation version of it that's important for horses gaining a new pacing gait. So it's how they walk, it's a new behavior. And this CEBP beta transcription factor is duplicated and assumes a new function during the development of the placenta and the split for placental mammals. And so that's an example of how the evolution of transcription is important for changes between species. So how does it all work? This is going to be a review for some of you, probably all of you. But we now know that 1 or 2% of the genome encodes for protein, and we know where it is. And estimates vary, but maybe up to half of the rest of the genome encodes regulatory DNA. And I'm highlighting some of that in these pink boxes. And these are classically called enhancers. They're regions of regulatory DNA that can be quite far away from their target genes. And the grand goal of genomics is this: how do we predict gene expression patterns in specific cells at specific times? And we now know that enhancers and transcription factors are really critical for this process. And solving this question, I believe, is really about solving three sub-problems. The first problem is, where are the enhancers? And this is the problem that has seen amazing progress in the last 20 years. So 20 years ago, we had about 100 enhancers. Now, thanks to functional genomics and consortia like ENCODE, there are millions of predictive enhancers, and tens of thousands of these have been validated. And people are also making progress linking enhancers to target genes, which can be quite tricky. So this problem is not solved, but good people are working on it, and they've made amazing progress. If we zoom into any individual enhancer, what we see is that it's a collection of transcription factor binding sites, and these are proteins. And so the second problem in this space is, what transcription factors bind each of these enhancers? And again, that's been the focus of a lot of attention. A lot of people work on this, and they've made tremendous progress insofar as now we can design a transcription factor to bind a specific DNA sequence. And that's pretty cool. But even if tomorrow we knew where all the enhancers were, and we knew exactly which transcription factors bound each of these enhancers, we still wouldn't be able to predict gene expression from sequence. And that's because we also need to understand, how do these transcription factors control gene expression? How do they recruit these sometimes large protein complexes that actually interact with the polymerase and make mRNA? And this is the problem that I have chosen to focus on, both as a postdoc and to launch my lab on. As a graduate student, I focused on enhancers, and I felt like a major limit was our understanding of transcription factors. So what is a transcription factor? It's a protein that has two jobs. It has to bind DNA, and it has to bind other proteins. We frequently draw them as a blob, and I'll do a slightly better. I draw it as two blobs. This is the DNA binding domain. It recognizes a specific DNA sequence. And then the rest of the protein has what we call effector domains. And these effector domains come in very different flavors. They're activation domains that activate transcription. They're repression domains that repress transcription. Dimerization domains, ligand binding domains, nuclear localization signals. I'm broadly interested in all of these, but my postdoctoral work has been focused on activation domains. And activation domains bind coactivator complexes. And they're operationally defined. They're a chunk of protein that I can cut out of the native transcription factor, attach to a new DNA binding domain, and it'll then activate a gene that's bound by this new DNA binding. And that's actually been a limit in the field, that they're operationally defined. So it's useful to compare DNA binding domains to activation domains. And that's because we know a lot about DNA binding domains. They're highly conserved. This is position along the protein. This is conservation of the GAL4 DNA binding domain across 34 yeast species. This is data. Some people think this is a cartoon, but it's data. And the activation domain is much more poorly conserved. In addition, DNA binding domains are structured. When we crystallize them, we get these beautiful structures. But activation domains are not. They're intrinsically disordered proteins. They are frequently these random coils in solution. Sometimes they will fold when they bind their cofactors, but they have high disorder. And this is a disorder predictor run just on the second activation domain of the GAL4 protein. This is the same region. And if we run this same disorder predictor on the DNA binding domain, we see that it has a much lower predicted disorder. And it is indeed structured. So I'd like to show this picture. This is a crystal structure of the interferon beta enhancisome. This is a triumph of structural biology and transcription. It's 8 transcription factors bound to a short enhancer. And they bind in a highly cooperative way. What I need to emphasize is that the only thing we can see here are the DNA binding domains. The activation domains are necessary for activation, but they don't show up in the crystal structure. So conservation and structure, low disorder, have given us really good models for predicting DNA binding domains directly from amino acid sequence. we have no similar models for activation domains. So one of the long-term goals is, can we build models to predict activation domains directly from amino acid sequences? Why do we want to do that? Well, we currently have great models for DNA binding domains, and as a result, that's how we organize transcription factors. These are the 1,600 transcription factors in the human proteome, organized by DNA binding domain. The black bar indicates that we know what DNA sequence these proteins bind. And this has been hugely successful and hugely useful. If we look at the rest of the protein for each of these 1,600s, the picture is a little bleaker. So this is the number of effector domains. In the original paper, they call it the number of non-DNA binding domains. And I want to emphasize this first column, which is over 600, so over a third of the transcription factors in the human genome. The only predicted domain is the DNA binding domain. We know it binds DNA, but we don't know even, is it an activator, is it a repressor, is it both? And so there's a huge opportunity here to go back and search for activation domains and eventually repression domains on these proteins. Just to know, to first approximation, how they regulate transcription. These guys, these are well studied now. Even here in this 1 and 2 and 3 column, in many cases we don't know where the activation domain is. Based on sort of my analysis, for only about 23% of transcription factors can we predict the activation domain using something like PFAM. All right, and so that's just to motivate this long-term goal of predicting activation domains from sequence. And so that's the big picture question, where do we begin? So in my postdoctoral work, I've been focused on this question. What are the amino acid features of transcription factor activation domains? So a little bit of history, which Leighton, I think, touched on. The first model for acidic activation domains was based on this paper in 1987, where Junman and Mark Patashnian, they sheared the E. coli genome, they fused this pseudo-random DNA to a DNA binding domain, and they recovered a whole bunch of new activation domains. And all these original activation domains, the only thing they had in common was they had a net negative charge. And that led Paul Sigler to propose this acid blob negative noodle model. And so the first model for activation domains is they use their negative charge to electrostatically interact with the polymerase and activate gene expression. This paper is also important in the intrinsically disordered protein literature because it's one of the first cases where someone seriously argued that a protein does not need to be structured in order to be functional. I'm going to redraw this cartoon like so. So negative charges are important for activity. That's the first one. A few years later, people started doing site-directed mutagenesis on these acidic activation domains. And what they found is that the hydrophobic residues are actually much more important. So the second model was it's not really negative, it's not negative charge, it's hydrophobicity. The third model came from people that couldn't crystallize these activation domains, but they started doing NMR, and what they found is that these regions frequently fold into alpha helices when they bind their co-activators. And so coupled folding and binding can be really important for their function. So we call this the disorder to order transition model. And then finally, when I started working on this, the ascendant model was that activation domains were going to be just like any other protein-protein interaction. It's going to be a short, linear, hydrophobic motif embedded in a disordered sequence, and that's going to be sufficient for activity. And I want to emphasize that each of these models is supported by conflicting data. There are a few constructs that support them, and at least 1 or 2 interesting counterexamples. And so we basically said, let's collect some more data and figure out what's really going on. And so in order to collect a lot more data about activation domains and make mutations to understand how they function, we developed a new high-throughput method in yeast. And so this is the yeast method. There's a synthetic transcription factor. It has an M-cherry protein, so we can measure the amount of protein in the cell. It has a DNA binding domain from mouse that does not interfere with the endogenous yeast gene regulation. It has this estrogen response domain. This is a human domain from estrogen receptor. It keeps the protein in the cytoplasm until we add a small molecule, beta-estradiol, which is an estrogen analog. And then the protein translocates into the nucleus when we add this drug, and that's so we can turn the system on and off. And we fuse to the end of this a library of designed activation domain mutants, variants. And I'll tell you in the next couple slides how we design those variants. And then we have a barcode sequence in the 3' UTR to uniquely identify each activation domain variant. We integrate this into the yeast. Each cell gets one and only one construct. The yeast makes the protein, enters the nucleus, and it can activate this GFP reporter. And so this is what the GFP signal looks like. Without the estradiol, we have a baseline amount of GFP. This is a histogram. And as we add the estrogen, the protein goes into the nucleus, it activates the GFP, and we get this shift. When we integrate the entire library of variants, we do 6,000 all at once, we get a distribution of fluorescence values. We do this all in a pool, one mixed pool. And what we actually look at is the ratio of GFP to M-cherry. So if it's a strong activation domain, it makes a lot of GFP per M-cherry molecule, and it'll have a high GFP M-cherry ratio, and that variant will be over here. If instead the variant is inactive, it'll make very little GFP. It'll still have M-cherry, so it'll accumulate over here with a low GFP to M-cherry ratio. And so all the mutants, all the variants are spread out in this distribution. And then we use fluorescent activated cell sorting to sort the cells based on this ratio. And then we extract genomic DNA from each of these pools and sequence the barcodes. And so then the barcodes have a distribution across these bins, and we take a weighted average. And so highly active ones are over here, inactive ones are over here, moderately active are in the middle. And so you can take this weighted average and you can compute an activity for every variant. The assay is reproducible, it's quantitative, has a pretty good dynamic range. We've figured out how to make the dynamic range better, and it works. But to convince you that it works, that's to remind me to tell you, in the first experiment we did 6,300 variants. So what did we decide to work on? So we focused on one of the most famous yeast activation domains, and this is from GCN4. GCN4 is a starvation response transcription factor. This is the first identified activation domain in it. I've highlighted the negatively charged residues in red. In the center is this motif, WLF, which is critical for its activity. It's an instance of this 5XX55 motif, which is also seen in mammals, and many activation domains have this motif. If you look at conservation, you pick up the motif and a proline. And then site-directed mutagenesis found the primary motif, but I had also pointed out that there's a secondary motif that's important for its activity. And then NMR experiments in biochemistry had shown that the middle section of this folds into an alpha helix when it binds its key cofactor, Gal11, which is the Med15 module of mediator. So to convince you that the assay is working, I'll show you the controls. These are all the variants that were in the literature when we started, so we included them 10 times each in the library. Everything's normalized to wild type. So these were the mutations that were expected to decrease activity, and they do. These variants were expected to not change activity, and they don't. And then these two variants were expected to increase activity, and they do. So this convinced us that the assay is basically working. And so now we're ready to do an experiment. So what did we decide to do? We wanted to test these 4 conflicting models for how activation domains work. And to test these models, we designed a set of mutations to test the models one at a time. So I'm starting with wild type here. This axis is net charge. This axis is net hydrophobicity. And this axis is predicted disorder. So these blue variants systematically vary net charge, and they test the importance of charge. The green variants vary hydrophobicity, and that's to test the role of hydrophobicity. And the light blue variants vary predicted intrinsic disorder. And I hope you'll appreciate that we worked very hard to design mutations that vary these properties one at a time independently. Separating charge and hydrophobicity isn't that hard, but separating hydrophobicity and predicted disorder is actually difficult because they're highly correlated in natural proteins. In addition, underneath the pink dot, there are 1,200 variants that all have the same charge, hydrophobicity, and predicted disorder that are shuffle sequences. And I will come back to those at the end because they become very important. These variants are complex. They each have 4 to 6 substitutions. In total, we tested 6,300 of them. Next, I'm going to recolor this graph, but the color is going to indicate the activity of the variant. So we measured all the variants in parallel in an experiment. Its position will still indicate hydrophobicity, charge, and disorder, but color will be activity or strength. Here is the color bar. Wild type is purple, inactive is nearly black, and highly active variants are gold. Here are the data. Let's walk through this. These variants change charge, and they're not highly correlated with activity. This set of variants changed net hydrophobicity, and they're also not well correlated with activity. This set changed predicted intrinsic disorder, and it looks like there's a correlation here. So we're going to focus on that first. So this is replotting just the vertical axis, predicted intrinsic disorder using the IUPRED algorithm and activity. And if you do an ANOVA, 57% of the variance is explained by this parameter. We got really excited about this because we thought what was happening is that these variants, which have lower disorder and higher activity, might be pre-folding the central alpha helix and be reducing the entropic cost of binding the coactivator. That's not what's happening. But in testing that idea, we arrived at what really is happening. And so now I'm going to describe simulations that we did of these variants and simulations of these variants and wild type. And by contrasting these two groups of simulations, I'll tell you how we think these systems work. All right. So this was too many mutations to go do NMR on all of them. So we wanted to get a sense of the structure, the ensemble. So we turned to simulation. And working with Rohit Papu, we've been doing these all-atom Monte Carlo simulations that capture the ensemble, the conformational ensemble. There are no dynamics. And as we simulate this protein, it wiggles around. It's a disordered protein. So blue is the N-terminus, red is the C-terminus. The middle is this motif. And if you take many snapshots and align them, it looks like this. It looks like a ball of spaghetti. It's not that useful by itself, but it's useful to compare the variants to each other. And this is one of the first highly active variants we simulated. And as you can see, the middle, the motif, is folding beautifully. And so we thought, yes, maybe it's pre-folding. But as we simulate more and more variants, the effect completely disappears. So this is the set of high disorder, low activity variants. This is the set of low disorder, high activity variants, and there's no difference. So the first 10 we did, the first few simulations, there was a difference. As we simulated those same 10 more, same variants, we did more simulations of those same variants. And as we added more variants, both of them regressed to the mean. Importantly, all of these variants are still disordered. Like we tried really hard to engineer disorder using this algorithm, and we really couldn't move the needle very much. So what is going on? This is a complicated plot, but it holds the answer. On this axis is the radius of gyration. That's a way of summarizing the size of the conformational ensemble. If it's expanded, it has a large radius of gyration. If it's collapsed, it has a small radius of gyration. Each point is an independent simulation. So these are 18 simulations of wild type. On this axis is the accessibility of the aromatic residues. It's whether the aromatics are sort of displayed and open versus whether they're collapsed and hidden. In red are the highly active variants. In blue are the inactive variants. So now there's 50 red sequences, each simulated 10 times, and 50 blue sequences, each simulated 10 times. And I'm going to show you an example because it's a little easier. This is a highly active variant that's collapsed. But as it collapses, it keeps this orange aromatic residue on the surface. And so that residue is exposed, even though this whole sequence is collapsed. This variant is expanded. But as it expands, it's waving around these two aromatic residues. So this one also has very high exposure of its aromatic residues. And by contrast, this is a low activity variant. And its aromatic residues are all in the middle, away from the solvent. It's very expanded, but the aromatics are less exposed. And so this is what's really, we think is really going on here, is that the active variants are keeping their aromatic residues exposed. We know the aromatic residues are important for interacting with the cofactors. So the active ones keep their aromatic residues exposed where they can interact. The inactive variants hide their aromatic residues, and so they're less active. They have stronger, weaker, they're less able to interact with other proteins. And so let's go back to this correlation between predicted disorder and activity. What's really going on here? Well, we also found that if we just count the number of these 5 amino acids, tryptophan, tyrosine, leucine, phenylalanine, and methionine, we can explain more of the variance along this axis than the predicted disorder. And when we add the predicted disorder on top of this, the marginal change is very small. And so we interpret this as this disorder predictor happens to count these amino acids very well. And that's why it's correlated with activity, not because it's actually capturing the disorder variation all that well. And so these two pieces of data argue that it's really about aromatics controlling activity, and so we then zoomed out and thought about it in the context of the full data set. So now I'm going to look at all the variants that change sequence, and these will be box plots of the number of tryptophans, the number of phenylalanines, and the number of tyrosines. These are cartoons of structures to remind you what amino acids look like. If you're like me, I'd actually forgotten what tryptophan looked like when I started this project. And so as we add tryptophans, we increase activity, and we see something similar for phenylalanine. It's not quite as strong, and tyrosine is similar, but also not quite as strong. And so the first approximation, the strongest signal in the data, is that the number of aromatics controls activity. So the second conclusion is that the number of aromatic residues is really critical for activity. But aromatics are not enough. And they're clearly not sufficient, because if you just put a cluster of aromatics together, that's a great way to nucleate folding of a protein and not a particularly great way to make a protein-protein interaction. And so to look at what else is important, I'm going to focus on the set of mutations that were designed to test the sufficiency of the short linear motif to create an activation domain. So the motif is, sorry, you can see that, WLF. And there was one instance in the literature where if you put this motif into a disordered region, you can create an activation domain. And we wanted to test how general is that. And so to do that, we designed these mutations that all fix the motif and then shuffle the rest of the sequence around the motif. They all have the same composition. They all have the same net charge, the same hydrophobicity. We filtered them to have a less than 1% change in predicted disorder. And so it's just changing the sequence context around the motif. How does this affect activity? Well, to my surprise at least, these variants span the entire dynamic range of our assay. This is wild type, plus and minus 1 standard deviation. These are highly active variants. These active variants are basically dead. They're completely dead. And so half the time, these sequences are as active as wild type or more active. So half the time, the motif is sufficient to create an activation domain, but clearly not all the time. And to get the sequence features, we compared the top 100 to the bottom 100. And the answer is that charge is important. Here's the linear sequence along the protein. The WLF motif is fixed. It's the same in all the sequences. And then what I'm summarizing is the fraction of the top 100 variants that have a negative charge at this position. And this is the fraction of the bottom 100 variants that have a negative charge at this position. So the 18 out of 100 have a negative here, 38 out of 100 have a negative here. And if we just look at the active variants, it doesn't look like much. But when we compare them to the low activity variants, something different happens right here. The high activity shuffle variants have a lot more negative charges around the motif. The low activity variants have many fewer negative charges right around the motif, particularly here. And instead, they've pushed their negative charges out here to the C terminus. Every one of these sequences has the same number of negative charges. It's just where they are. And when we look at positive charges, we actually see the opposite signal. So this means that this motif needs a negatively charged context in order to be active. And so that's the third conclusion of my talk, that the motif needs to be surrounded by this negative charge. And you can also say that as negative charges create a permissive context for the motif to be active. So can we use this to predict new activation domains? Are these rules general? There are 4 instances of the WLF motif in the yeast transcription factors. GAL4, GCN4, which I've been telling you about. That's our positive control. GAL4 is another transcription factor, and it has this motif, but this is not an activation domain. And that had previously been shown. It was actually in the literature as one of these, like, why isn't this an activation domain? And the other 2 are STB5 and MOR1. And this one is at the end of the protein, so that's why it's not centered on the motif. And so based on these context rules, we learned that negative charge and disorder seem to be important for activity. We predicted that this one would be inactive, and this one would be active. And this is what the data look like. If there's no activation domain, there's very little change between gray and green. With an activation domain, the green shifts to the right of the gray. So this is with and without estradiol. This is a negative control. Very little change. And our predictions are borne out. STD5 is not an activation domain. WAR1 is an activation domain. So we got really excited about this. And we really wished there were more instances of this motif so that we could keep going, but there aren't. And so now I want to take a moment to address sort of the question I posed at the beginning, like, what's it going to take to predict activation domains from amino acid sequence? And what we realize is actually 2 problems. The first problem is, given an activation domain, how do changes in the sequence of that domain impact function? And that's really a personalized medicine question, because there are activation domains that have mutations, and then patients arrive, and they have a new mutation in that domain, and does that mutation change the function of this protein? And most of the time, we don't know. So that's a useful problem to solve, at least in clinical setting. And so I'm not going to go into this, but I just want to say we did some basic machine learning, this to support vector regression, on the data we had, and we can explain the majority of the variants in the training set, and then we also left aside the orthologs, the sequences from other species, as a separate test set, and we do just as well in a test set. So that gives us hope that we will be able to design machine learning models to predict how variants impact expression in known activation domains. So that's around a known domain. That's a different problem of how do I go into the proteome and predict activation domains from scratch. And so going forward, I'm interested in that problem. And what we've realized is that that's going to take a much more diverse set of sequences. And we're going to have to build a family of models. Not all activation domains are the same, and we might have to build, just like for DNA binding domains, we have a homeodomain model, we have a leucine zipper model, we have a helix string helix model. We're going to have to build a family of different models for predicting activation domains. And we don't actually know how many families there are, so we don't know how many models there will be. Okay, this is the conclusion of the first third chunk, almost half of my talk. We have this unified model for how an acidic activation domain works. The acidity and the disorder keep aromatic residues exposed to solvent, and that way they're available to interact with the co-activator machinery. of the classic models contribute to this unified model, but none of them is sufficient. And so that's why we're arguing it's a unified model and not a totally new model. All right, at this point, some of you might be wondering, well, what about the fifth model? There's this new model, that liquid-liquid phase separation is explaining what's going on in transcription. These mutations were not designed to test that model. But I will say everything we're seeing is consistent with that hypothesis. And I'd be happy to talk more about that, but I don't have that much more to say in this talk. All right, and so at this point I get a few questions about this model. So if it's really all about exposure of aromatic residues, why is it negative charges? Why can't it be positive charges? And there are at least three answers to this question. The first is empirical. The coactivators are positively charged. So this is Gal11. This is the shallow hydrophobic group where the GCN4 activation domain binds. And the blue are the positively charged regions. The coactivator is positively charged. And in some activation domain co-activator interactions, these charge-based interactions do contribute to binding. They don't contribute a lot, but they contribute a lot. The other explanation is that the DNA is negatively charged. And if the activation domain were positively charged, then it would be sticking to the DNA. And it would, you get a lot of non-specific binding of the transcription factor to the DNA. And by making it negatively charged, it actually sticks up and sticks out and is even more exposed. And then the third case, which is quite interesting, is in some cases, the activation domain can turn around and bind the DNA binding domain, actually compete with a non-specific DNA binding. And that's a charge-mediated effect. All right. Do activation domains actually need to be disordered? They all are disordered, with one interesting exception. And here, people have argued that these disordered domains fold into a different structure when they bind each coactivator. So this is the P53 activation domain. This is the TAZ2 coactivator complex binding partner. This is the TAZ1. And in this case, this alpha helix points down, and in this one, it points up. And these are these two domains are duplicated. They're very similar. This is the equivalent binding surface, but the domain is going in the opposite direction in these two cases. And so people have argued that in order to assume a different fold when binding each cofactor, the activation domains need to start disordered. The other argument, the other reason we're pushing the other possibility for why it's useful for these activation domains to be disordered is because it's entropically expensive to keep aromatic residues in solution all the time. And it might be much cheaper and easier and have a larger evolutionary space if it's only exposed part of the time. And that's easy to achieve in a disorder. And then the final question is, why aren't activation domains just aromatic residues and negative charges? Is this a functional activation domain? And the answer is, this is a functional activation domain. It's actually very strong. Madame Babu's group tested this in England. And here, I think the answer is in regulation. That this sequence is strong, and it's too strong, and it's strong all the time, and it can't be controlled. And the additional sequence diversity allows these activation domains to do different things. And that's what I'm going to tell you about in the next part of the talk. This is just repeating everything I just told you. Activation domains bind positively charged coactivators, and they fold differently when they bind their different coactivators. But I want to dig into this hypothesis, that the reasons these activation domains have sequence complexity is to enable regulation. So GCN4 is an amino acid starvation response transcription factor. And its primary means of control is that the mRNA is always present, and it's translationally repressed when amino acids are present. But during amino acid starvation, the mRNA gets translated, the transcription factor goes into the nucleus, and then activates the amino acid synthesis genes. We had a completely synthetic system with no translational control. And so the synthetic transcription factor is in the nucleus regardless of whether the cell is starved or not. And so we expected activity to be the same in both cases. And that's not what we see. What we see is that the activity of the activation domain is 1.7 times stronger under nutritional starvation conditions. So in the nucleus, where this protein has evolved to function, it is a better activator. What do the variants look like? So now there are two phenotypes. This is activity in media with amino acids. This is what I've been telling you about the whole time. And on this axis is the fold change under amino acid starvation. Here's wild type. It's not that strong. Everything's normalized to wild type. But it's not that strong. And it has a 1.7-fold change. Here are all the data. Let's focus on the histogram for a moment. 68% of our variants are stronger than wild type in media with amino acids. This really confused us. Why is it so easy to make a mutation that makes this protein stronger? And the answer is in the responsiveness to starvation. Very few of our variants are more responsive to starvation. And so what we believe is that the GCN4 activation domain has been selected to be weak and responsive, and that there's a trade-off. If you look at this, there's a negative slope here. It's difficult to build a protein over here that is both strong and highly responsive. This is our detection limit for this responsiveness. In the paper, we called it induction. So these guys are not well measured, but these guys could be more induced than they aren't. And if we look at the orthologous activation domains from other yeast species, we see that they follow this trade-off. Many of them are weak and inducible. And then this set over here is interesting because this is from a distant plated yeast, and the activation domain looks different and is clearly a lot stronger. And so there's reason to believe that once this protein is so far away, it might actually be regulating different targets or be used in a different system. All right, so to conclude the next section, the GCN4 activation domain is weak and inducible. And then I think these trade-offs are actually going to be pervasive. And that's part of why these activation domains have high sequence complexity, and may also be why they're evolving so, because they are evolving. So I just told you that activation domain activity can change with cell state. That means in principle, it could also change with cell type. So an activation domain that's important for making this bristle ciliated cell, if it gets misexpressed in this fibroblast-like cell, it might have a lower intrinsic activity because it's not matched to the cofactor, coactivator environment. And that's really cool because it creates barriers to reprogramming, and it also enforces canalization of cell fate in the developmental setting. And so going forward, I'm interested in cell type specific activation domain activity. All right, so to conclude the yeast section of the talk, I told you about this high throughput method I developed to study activation domain variants. We came up with this unified model that activation domains need to keep aromatic residues exposed so that they're available to interact with coactivators, and they use negative charge and disorder to do this exposure. And then we have this functional trade-off between baseline activity and induction, or responsiveness to starvation. So in the last few minutes, I'm going to tell you about some new data that is rebuilding the entire system in human cell culture. So it's trying to build a very similar high throughput assay in human cell culture. Some of you might ask, why go through the trouble of building this again in human cell culture? It's a lot more work. And there are a few reasons. The most interesting reason is that there are parts of transcriptional activation that are missing in yeast. So my favorite is that yeast don't have enhancers, meaning there's still regulatory DNA. All the regulatory DNA is at the promoter. In addition, I told you there's this great opportunity to look for more activation domains on the human transcription factors that probably have them, but we don't know what they are. And then finally, there's a clinical reason, which is that patients are arriving in the clinic, we're sequencing their genomes, and we're finding novel missense mutations in the transcription factors, and we have very little ability to predict how these new mutations impact function. So they get labeled as variants of uncertain significance. And there's this growing movement to use cell-based functional assays to help nudge these variants into likely pathogenic or likely benign status. And that's useful for clinicians and geneticists. The main challenge of replicating this system in human cell culture is we want to introduce one and only one variant per cell. And we want to introduce it all at the same place. And the reason we want to put them all at the same place is because in human cells, position effects are large. Position effects are you integrate the same gene in different places, and you can get 100-fold or sometimes 1,000-fold differences in expression. There are position effects in yeast. They're just empirically bigger in human cells. So I've been using this asymmetrics LOX-P system that a pair of graduate students in the Cohen lab developed. And the way this works is they've built this landing pad, which has these 2 LOX-P sites. We transfect in our library of activation domain variants. This part is basically the same as the yeast. And we use Cre recombinase to integrate these constructs at the landing pad. And this greatly reduces the cell-to-cell variability in expression of these proteins. We still use a GFP reporter. This time we're using a completely synthetic DNA binding domain that's been specifically engineered to have very little cross-reactivity with the human genome and to very strongly bind this reporter with 4 sites, mococula and nucleus. Again, we sort the cells based on the GFP level, and we sequence the barcodes from the cells. This time we decided to focus on 2 activation domains, and we picked the 2 activation domains from the tumor suppressor transcription factor P53. We picked them because they're similar to GCN4. They both have a motif. It's a phi XX phi motif. If you're paying attention, this is an anagram, the one we had in yeast. The conservation shows that hydrophobic residues are the most conserved residues, but if you look at the different sequences, they are always negative. There was genetic evidence identifying the most important positions, and the central regions fold into beautiful alpha helices when they bind to some of their cofactors. This table summarizes the mutations we made. We started with classic mutations in the literature. This is so we could benchmark the assay for method development. And this time we chose to do a deep mutational scan. So a deep mutational scan is you take each position in the protein and you mutate it to all other amino acids. So it's 19 times the length. These have become very popular specifically for testing all possible variants so that you can prioritize these variants of uncertain significance. We were also interested to do all the mutations so that in the future we can understand what are the most informative substitutions. We included in this all the variants we could find in XAC Nomad. This is enriched for variants that are common and presumably don't have deleterious effects. And then we used all the variants in Cosmic, which is a database of mutations found in cancers. I'm actually not going to focus on this set, and that's because the first version of this assay, we didn't have very good resolution to measure the effect of point mutations. And since then we figured out how to do this. We simply need more barcodes per variant in order to get a better measurement and reduce our error bars. So this is a disordered sequence. Most substitutions in this disordered sequence actually have a very small effect on activity. And that effect is frequently smaller than our measurement error. And so I'm going to focus on the hand-designed variants because they have a much larger effect size. And so it's very easy to detect how they're impacting the protein. And that's what we can see. All right. Those variants were designed to test this unified model for activation. And so this model makes a series of predictions. The first prediction is that removing the motif should decrease activity. So this is activation domain 1. This is wild type plus minus standard error of the mean. And double mutants in the motif decrease activity pretty clearly. Activation domain 2, the effect size is smaller. The second prediction is that if we remove the negative charges and start to replace them with positive charges, we should start to lose activity. And this is what we see. So activation domain 1, activity clearly starts going down as we go first neutral and then to positive. And similar for activation domain 2. What about adding negatives? What happens when we add negative charge? The prediction is that this should increase activity. Here we start to see something slightly different between the two domains. Activation domain 1 nicely increases activity. Activation domain 2, it's the same or slightly stronger. And this difference is for two reasons, we think. So first, it's that activation domain 1 is phosphorylated in 4 positions, and 2 of those positions are known to increase activity. And the second is that activation domain 2, if you look carefully, it's actually more negative and shorter than activation domain 1. So if you renormalize the data based on length as a net charge per residue, it's actually all the way over here. And we may be saturating. It may start out saturated for the effect of negative charge. What about the opposite? The final prediction is if we add aromatic residues, we should increase activity. And that's what we see. So for activation domain 1, adding aromatics generally increases activity, but the effect's not quite as robust. As activation domain 2, we're adding aromatics robustly and consistently increases activity. And here what we think is going on is there's a balance between the acidity and the aromatics. These activation domain 2 is more negative. And so we think if you take an extra aromatic and you put it anywhere in the sequence, it's able to display it and keep it exposed and become stronger. Versus this one doesn't have enough negative charge. So I'm going to draw that as a seesaw, just to be more clear. Activation domain 2 has an excess amount of negative charge, and so adding aromatics is an easy, robust, consistent way to make it stronger. Activation domain 1 doesn't have as many negative charges, and so adding negative charges robustly and consistently increases its activity. And then adding more aromatics doesn't necessarily increase activity because it's actually limited for negative charge. And then just to show you very quickly, oh shoot, I'm running out of time. Does this generalize to even more activation domains? In the last experiment, we decided to double down on this negative charge aromatic, and we looked at 5 more strong acidic activation domains. And we systematically varied charge and aromatics. These are the effects for charge. So these are the 5 domains. This is charge. And these 3 have the same strong effect as P53. As we reduce the negative charge and make it more positive, we lose activity very quickly. These 2 are a little different. The negative slope is not nearly as strong. And so this might reveal that there are different kinds of acidic activation domains. And when we look at aromatics, we're actually starting to see something different now. And this is brand new. We don't fully understand this, but I wanted to show it to you. So for these 3, adding aromatics is no longer increasing activity. So we're going to have to figure out what's going on there. I'm not totally sure. But these 2, it's totally flat. And it's flat for both charge and aromatic. So I'm starting to believe that these 2 domains are a different class of acidic activation domain that's working differently from this GCN4 P53 axis. All right, so in conclusion, we have a system for integrating thousands of variants into the specific location in human cell culture. We've scaled this up so we can get 60 or 80,000 integrations per experiment. It takes some work, but we can get there. We've measured many activation domains in human cells. And then for P53, we're seeing this acidity-aromatic balance is important for their activity. And we also have tantalizing evidence that at least 2 other acidic activation domains are working differently. And so to remind you, we currently organize transcription factors based on their DNA binding domains. And one of the long-term goals of my work is to find all the activation domains and then to reorganize them based on how they activate transcription, and to impose this orthogonal organization that will be useful, either for studying developmental regulatory networks or studying different modes of transcription. And the dream is that if we really understood how transcription factors control transcription and what types of transcription factors need to be present to robustly activate a gene, we might be able to go backwards and predict where transcription factors are bound in the genome and where the enhancers are. And that would be useful in modern systems. So to give you a flavor of what's next, the first goal is to find new activation domains and define their motifs, and that's in service of this question of what is an activation domain, and with the goal of predicting activation domains from amino acids. And prediction isn't enough. We actually want to understand how they work. And so I'd like to characterize the different kinds of activation, and to use different clever reporter assays, most of which are already in literature, to subcategorize different types of activation. And that's to get at this, how do activation domains work? What are the different classes? How many classes are there? But really, both of these questions are in service of the bigger question, how do transcription factors work together? And I'm going to focus on how the activation domains work together by looking at this synergistic activation. So if two activation domains strongly and synergistically activate expression, that tells us something different about how they're activating transcription. So how do activation domains work together? How do transcription factors work together? And with that, I will thank some people and then take questions. But most of all, I need to thank Barack Cohen, this guy, who's been a fantastic postdoctoral advisor. I arrived in his lab, told him I want to repurpose your experimental system for studying regulatory DNA to study protein, and he let me do it. But he also sent me to go talk to Rohit Papu, which is this guy, who's an actual expert on proteins, and he's kept us honest and helped us do better experiments in protein space. Support centers, Mo Cockfield designed the DNA binding domain for the human assay, and of course funding. And thank you all for listening. I'll take questions.
So is the cofact, do you know if the cofactor is always the same when you introduce these mutations? And I guess the second part of that is like how conserved are features of these cofactors?
The cofactors in general are much more conserved than the activation domains. We don't know definitively that it's the same cofactor, but Steve Hahn has done a panel of mutations and shown that it is still the same cofactor in that case. And so we're not moving very far in sequence space, so the reasonable expectation is that it is still the same cofactor. And there's been there's a group in England that did some MD and showed there that mutations that were known to increase activity sort of lengthened the binding interface with the same cofactor.
I'm just curious if you know anything about the molecular relationship between the amino acid starvation and response of the activation domains, and how some are better and some are worse.
There are sequence features that are enriched in the best responders. I didn't talk about that, but it's actually the negative charges in the middle are the most important for that. We were looking to look for like a post-translational modification signature. We couldn't find that. There's a phosphorylation site, but it's mutated the same, just as often in the high responders and the low responders. Our favorite hypothesis is that a coactivator complex either changes activity or abundance, but we haven't nailed that down.
I think the system that you were showing in humans is really just, so the lack of nicety in like the results compared to the yeast studies, do you think that's really just a matter of increased complexity in regulation coming from a human system while using a motif that's similar between the two? So do you think there's other factors?
At this point, I think it's mostly our measurement error is a lot larger. We're not measuring them as well in the human system. So I'm not prepared to say that it's actually more complicated, but we'll see. I might be wrong. Or we've stumbled on, or we're working in. There's a distinct possibility that we've suddenly started working on a different type of activation link. So I didn't emphasize, I should have, that the model for GCN4, the fact that it explained anything about P53 was actually really exciting to us, because those are pretty different. And so the fact that it's even explaining some of these other activation domains is pretty cool. But it's not going to explain all of them, probably because they're working.
I don't think you ever said what the significance of aromatics is as opposed to any other hydrophobic, especially since in some of the motifs you do have solution.
So it's mostly a quantitative effect. Tryptophan, then phenylalanine, then tyrosine. Leucine it works, but its effect size is smaller. Methionine works, but it has a very small effect size. Isoleucine and valine, no effect on activity.
Any idea why? You like iron traction or something?
No, I don't have a good idea why. And in P53, isoleucine, but in GC4 it's not.
And I was trying to remember what the role of the negative charges was, it's just to stick the aromatics out.
That's what we think is the main reason.
So that would make sense if this thing wanted to be really good binder, but does it ever dissociate from its cofactor on it?
So it's incredibly dynamic. These interactions are not that dynamic, they're very dynamic, short-lived.
So have you considered if the negative charges could be there for some kind of evolutionary negative design to prevent it from blocking onto other things and just aggregating?
Sure. I think in particular it's keeping it away from DNA.
But it could also aggregate with itself if you have, if you need to make polyaromatics.
Yeah, for sure.
Um,
And keeping the aromatics in solution.
Also, do you have any idea, you ran the sequences through an intrinsic disorder prediction? What does that do? Because there's so many of these programs that you know.
Yeah, this is one of the ones that takes a sliding window, it has an energy matrix, it's trained on some few hundred PV structures. I mean, in our hands, it mostly counts the aromatics.
Because you could also think of running it through something that predicts secondary structure, like chatapasma.
Yeah,
Because most of these
that would
are-
probably be worth doing. I ran it through Agadir, and I have no correlation between.
There are other programs that predict IDPs based on propensity for secondary structure. I think it's called CORES.
Yeah, there's at least 4. I'm using the one that was popular in the Papu lab when I designed these.
Also for that PAPU sequence, you showed WD, WD,
Yeah.
WD. That'd be an awful Helix scanner for just period installation.
That's true, but it's a very strong activation. So that might be one that doesn't actually need to be a helix to be active. So that's a great point.
For your assay, in terms of the yeast, what was your variability between samples? What did that look like?
Oh, it's very low.
Very low, okay.
Very low.
And then for the humans as well, what is this, like your FTSE signal, the spread? I'm just wondering how so when you're getting your FTSEs, are you getting these nice defined peaks for each individual population? Because you don't correlate it to the proportion of.
Sorry, ask that again?
So for the human, right?
Yeah.
So you're correlating it to GFP signal, right?
We're using the GFP signal to sort the cells,
Mhm.
and then we're extracting the genomic DNA from each sample, sequencing barcodes.
So within each sample, are you getting nice defined peaks where you would expect? You're not seeing a large variability between your FTSE signal in the flow cytometry.
We should talk more because I'm not totally sure I know what you mean. It's all one big population, so the
Mhm.
signal is completely continuous.
Yeah.
And we're just drawing bins somewhat arbitrarily.
Okay.
So that we get the same number of cells in each bin. That's how
Okay. And within your bins, do you find that kind of correlates to the, let's see, the activator that you put in? Like, are you getting populations within those bins, or are you seeing kind of each sample spread all over the histogram?
Something in between. So we see like so for a strong variant, there will be multiple integrations. They'll each have each independent integration will have a slightly different histogram. The strong ones will be enriched on the strong side. There'll be an outlier that's very weak. And even for the weak ones, we'll have, histograms they'll be weak. So I'm doing this for me, not for you. It's reasonably well behaved. And the more barcodes, the better. So if we want to measure the effects of point mutations going forward, we're going to do that with 25, 30, maybe 50 barcodes, meaning replicates per sequence. If we're just screening for large effects, then we can get away with 5.
So one of the things that struck me as interesting that you said was that you actually don't know how many clusters or families of activation domains there might be. So I'm just trying to get my head around how you might estimate what you think you might find in the number of types. I mean, have you tried to extend your model across all transcription factors and see what percentage it might account for?
I mean, we did try that, but we want to do it I can look at acidic activation domains and see clusters of aromatics surrounded by negative charge, like in the list of 50 or so domains I've curated from the literature. And so I think maybe that's how they're working. But until we go in and really test them, I don't want to put too much money down on the table. I will talk about this a lot on the chalk talk, but there are like documented activation domain promoter interactions. So like some of them do better at TATA promoters and some of them do better at TATA less promoters. Many of them work at promoters. Some of them, not all of them work when they're far away from the promoter, sort of other means. Because the goal is to use assays like that to sort of subdivide phenomenologically.
So you showed there's a lot of positive charges towards the end of the least active domains.
Yeah.
In your modeling data, do you see those positive charge residues folding over by any chance?
To be honest, I haven't actually simulated any of the things that are that positive. But we should do that. I mean, it's on the list of things to do.
Quick question. What's the one exception to the rule that you mentioned? Disorder being an activation domain? Just curious.
I don't remember what that meant. You send me an email. I'll send it to you.
You mentioned the structure of the helix. Does it play any role in activation?
I mean, it's there. Many of these domains, if you put a proline in the middle, you lose 20 or 30% of activity. Does it need to be there? I would love to know the answer to that. I have not come up with the right experiment to test that. And some of them work if you add alanines to make the helix more stable to get stronger. So, jury's out.
How do you suppose your model fits into certain transcription factors that act as an activator for certain genes while also a repressor for other genes within the same
Great question. I love those proteins. I would like to work on them more. My personal belief is that most of those have an activation domain and a separate repression domain. And depending on the context, one or the other is masked or neutralized by other transcription factors nearby. So part of the reason for mapping all the regions that are sufficient to be activation domains and sufficient to be repression domains is to find the proteins that have these separable activities.
How do you control for changes in DNA binding when you're mutating the activation domain?
We make the assumption that it doesn't change.
Okay.
That's a reasonable assumption in this super synthetic system.
I know that it's relatively small activation domain, but you kind of hinted at there are changes to activation domains that can alter DNA binding that wouldn't show up in that fluorescence assay.
Absolutely. Those are in sort of endogenous, highly evolved systems. So like UBX, Sarabondos shows that as you truncate the protein, you get radically different changes in DNA binding affinity. And that's because the protein loops around and there's tyrosine-centered clusters that sort of compete with the DNA for the DNA binding domain. And P53 does something similar too. But I think that those are interesting secondary effects to this first order modularity. People used to say activation domains and DNA binding domains are separate modular things. To first approximation, much of the time they are. The exceptions are fast.